Assalamualaikum, we are from D1 and 3P1. My name is Kamatu Alika binti Ramlan.
My name is Nur Ashki bin Syaifuddin. My name is Alia Afika binti Zainal Abidin.
My name is Nur Amira Zulaika binti Muhammad Safri.
My name is Aina Bila binti Azri.
So our topic today is recombinant DNA technology. A definition of recombinant DNA technology is a method in which DNA molecules made in vitro with segments different sources or by combining genes by different organisms. Tools used by recombinant DNA technology are target DNA restriction enzyme, DNA cloning vector, host cell and modifying enzyme.
Next, I'm going to talk about the tools that use in the DNA recombinant technology. Target DNA is the sequence of DNA containing gene of interest. The next one is the restriction enzyme. The restriction enzyme is the enzyme which cuts the DNA molecules at a restriction site and produce short restriction fragments. Restriction site contain palindromy, where the same sequence of bases is found in the both DNA strands but in opposite directions.
It produces two types of cuts. The first one is TKN and It produces two types of cuts. The first one is TKN and blunt end. Examples of restriction enzyme is EcoR1. It produced by E. coli. Recognizes the base sequence of 5' GAATTC 3'. Makes staggered cuts within the sequence.
Another example of restriction enzyme is MA1. It produced by Serratia marcescens. Recognizes the base sequence of 5' CCC GGG 3'. The cut is made between the adjacent C and G.
The third tools that used in recombinant DNA is a DNA cloning vector. DNA cloning vector is a DNA molecule which can carry foreign DNA fragment into a cell and replicate in that cell. The characteristic of DNA cloning vector is able to accept foreign DNA, able to replicate freely in the host cell, and possess selectable genetic marker. The example of DNA cloning vector is plasmid, bacteriophage, cosmid, and YAC. Plasmid is a DNA molecule which can carry foreign DNA into the host cell and replicate there. Bacteriophage is a virus that infects to the bacteria. Cosmid is a hybrid vector which is combination of plasmid and G and cos G. YAC is a fragment of DNA from the human genome insert to the yeast cell.
This is plasmid, which is the example of DNA cloning vector. In plasmid, we have ampicillin resistant, origin of replication initiation, and lex.
Next, the tools used in recombinant DNA is host cell. Host cell is a cell which receives a recombinant DNA and allows the replication of recombinant DNA. The characteristic of host cell is able to receive recombinant DNA through transformation process, able to maintain the structure of recombinant DNA from one generation to another, and able to amplify the gene product of recombinant DNA. The example of the host cell is bacteria. Bacteria is commonly used as the host cell because DNA can be easily isolated from or introduced to bacteria. Bacteria cultures grow quickly and rapidly repeating any foreign genes they carry. Example of the bacteria used is the E. coli. And the last tools that use in recombinant DNA is modifying enzyme. The example of modifying enzyme is DNA ligase. DNA ligase join the foreign DNA fragments containing gene of interest and bacterial plasmid by catalyzing the formation of phosphodiester bond to form recombinant DNA.
The last part of topic of recombinant DNA technology is methods in gene cloning. So there are 5 methods in gene cloning. The first method of gene cloning is isolation from DNA. So here we have plasmid from the bacterial chromosome which we took out. Then the DNA donor which we took out the DNA. The next step of gene cloning is cleave or cut. So we have our plasmid and our target DNA. Our plasmid, their DNA is GGG and CCC. So they are both cut with the same restriction enzyme which is MA1. So they will be cut, straight cut and produce blood end. So the plasmid will be like this. Like this. Then our target DNA which is cut, also cut by the same restriction enzyme MA1, and they will insert into the plasmid. That's it.
Next, I will explain about the next stage after cleave or cut occur. The stage is insertion. Target DNA will insert into plasmid to form recombinant DNA, and recombinant DNA will join by DNA ligase. Alright, in this insertion stage, 2 probability will occur, which is recombinant DNA and non-recombinant DNA. The 4th stage is transformation and amplification. At this stage, recombinant DNA introducing to bacterial cells. Okay, the last stage of gene cloning is screening. At this stage, screening is to identify bacteria clone containing recombinant DNA. Blue-white screening is done. To identify it, place the bacteria on agar that containing antibiotic, ampicillin and sugar which is exhaled. It will incubate until the colonies grow. Take only white colonies and amplify it again.
That's